Good evening and welcome to the historic Wayne Theater Ross Performing Arts Center. Yay! We are so excited that you're here. Coming up at the Wayne Theater live on stage, our Studio Wayne kids will be doing performances of James and the Giant Peach on Saturday and Sunday, and Monday's performance has sold out, but still a few tickets for Saturday and Sunday. And then the following weekend, we have 2 performances of Matilda. Again, student performances, and tickets are on sale now for Queen, One Night of Queen. Andrew Duhon, the Celtic Angels, 2 performances at 4 o'clock and 7:30, so we still have tickets available. And we want to know what you want to see live on this stage or on film or through our signature speaker series. And we are so excited to have Science as our signature speaker series this evening. And to introduce our speakers is Dr. Tom Bensing.
Thank you so much, Tracy, and my welcome too to the Wayne Theater. Wayne Theater has been great as a partner in delivering these science talks. I also want to recognize some of our other sponsors, the Center for Cold Waters Restoration, the Virginia Museum of Natural History, and the South River Watershed Coalition. Sorry, they changed names. They were the South River Science Team. Now they're the Watershed Coalition. And official now. Max is in the audience, so we have an official 501c3 status. Tonight, before I introduce our speaker, I want to also identify our last talk, which will be in April for this season, and it's Southern Appalachian Brook Trout, a 50 million year journey to extinction, ? John Ross and Eric Hollerman, they're going to talk a little bit about the natural history of how brook trout arrived in Virginia and some of the genetics work that has been done to elucidate where they came from, essentially. So that'll be in April 11th on Tuesday. Tonight, our talk is about algae, and we solicit input from our audience regularly on topics that you want to hear. And so, as I said, we're closing out the season next month. If you have topics that you want to hear in the coming season, we'll start back in September. Certainly would like to hear from you about that. Tonight, the Friends of the Middle River came up to us and said, we'd like to do a program on algae in the Shenandoah River. And so I'd like to bring up Wayne Nolde to introduce the talk tonight, the speakers tonight. Wayne is on the board of the Friends of the Middle River. So please join me in welcoming Wayne to the stage.
Good evening. Appreciate y'all coming out tonight. As you should know by now, the name of our talk is When Green is Not Good, having to do with algae in the rivers. So first, just a little bit of background on Friends of the Middle River. Our mission is to promote the stewardship and enjoyment of the Middle River watershed through volunteer action, education, science, and outreach. Examples of our volunteer action include our river cleanups that we do each year. You just can't fathom the number of tires and the tons of trash that we get out of the river every year. An example of education is our work at Earth Day and Riverfest. And we've even done some education events at one of the local campgrounds along the river. Some examples of our science work, in addition to this evening, is the benthic and E. coli monitoring that our teams do. And examples of outreach include our newsletter, our website, and programs like this one tonight. So here you see a map of the Shenandoah River Basin. You can see it drains to the Potomac River. If you can see the pointer, it's right up there. And here we are. Friends of the Middle River is basically most of Augusta County. Of course, you got North River, that's the western part of the county, and you've got South River, that's the eastern part of the county, and logically, we're in the middle. And you can see down at the very bottom there's the Wayne Theater, which is where we are tonight. I will say this, please stop by our table out in the hall when the program is over so that you can sign up for our newsletter and talk to our people. This map shows the entire Potomac River watershed. Our guest speakers will discuss the differences in identification clues between nuisance algae and harmful algae that occurs in the rivers, Shenandoah River watershed. DEQ will review the last 6 years of nuisance algae monitoring efforts. Dr. Steffen will explore how harmful algal blooms, HABs, are hazardous to humans, pets, and livestock. Both speakers will also discuss how to report suspected algal blooms to authorities for future investigation. Our first speaker, Tara Wyrick, is the Regional Water Monitoring and Assessments Manager for the Valley Regional Office of the Virginia Department of Environmental Quality, and she will give you some additional information on her background. And our second speaker, Morgan Steffen, PhD, Associate Professor of James Madison University's Department of Biology, and she can also give you some additional information. So we hope that you enjoyed tonight's presentations, which will be followed by a question and answer session after the presentations. Thank you very much. And Tara, would you like to come forward? Okay.
We'll see. Hi, everybody. Thanks so much for coming tonight. I'm hoping that my mic is picking up. Thumbs up if you can hear me. Oh, perfect. Okay. I'll try not to blast you out. I have a projection voice. My husband tells me that I talk too loud sometimes, so I'll try to keep it in the neutral zone. But let's see. Make sure that this is working. There we go. Excellent. Well, thank you so much for the Friends of the Middle River for having us tonight and the Wayne Theater for hosting. As was mentioned, my name is Tara Wyrick. I'm the Water Monitoring and Assessment Manager for the Valley Regional Office. So I work with an excellent team of scientists. I'm lucky enough to work with really a team of experts, and the algae issue, as with a lot of our programmatic responsibilities, truly is a team effort. So here's just kind of a quick list of our staff, our full-time staff that's on our full-time pay band. And then we also hire 1, 2, sometimes even 3 wage employees, student summer interns, that sort of thing. So if you know of a college student who likes to get their feet wet and their hands dirty, tell them to give me a call. You can see down at the bottom here, there's a list of all of the different programs that our team does and the different types of monitoring we do. I'm not going to read it to you, but I think you can get the point that it's an extensive list there, and nuisance algae is just one of these different priorities. All right, so today we're going to just do kind of a quick overview of what is filamentous algae, what does it look like in our rivers and streams, talk a little bit about why this is a nuisance and how we've been dealing with it. We'll hopefully get pretty involved into how we collect the data. What does sample collection look like? What does algae look like in our streams and rivers? And then even talk a little bit about plans for this coming summer. So with that, we'll jump right in. Oh wait, there's some green stuff in our river. Does that look like something you want to get into? How about this? Is this something that you want your kids playing in? Is that something that you want your dogs to swim in? So there's a lot of algae in our rivers, and we really have to understand what's growing, where it's growing, and what's the line of too much algae before it starts impacting our recreational uses, right? Because I'm sure pretty much everybody, if I ask for a show of hands, everybody likes to spend a hot summer day in the river, either catching fish or angling or canoeing, or even just getting your feet wet like my daughter likes to do, or looking for crawdads is another fun activity. But really the question for us in this issue is, what is a nuisance level of algae to the point where it's impacting our recreational abilities? Oh, I had one more picture. There you go. There we go. All right, so first things first, we're looking at filamentous green algae specifically. So what is filamentous green algae? Well, it's chains of algae cells that link together, creating these long filaments, as you can see in this picture over here. They can be shorter filaments, just a couple of inches, or they can be several meters long. This, again, is green algae, so it's feeding off of chlorophyll. It does not have a vascular system, so we're not talking about plants that like to have their feet wet. We're really talking about algae specifically. It's non-toxigenic, which means that it does not produce toxins. It does produce a lot of oxygen, so the river water is usually very well oxygenated, where we're seeing a lot of algae. And it is a natural part of the ecosystem, right? It produces, or it serves as a food source. It's habitat for the different aquatic organisms. But what's the line between normal or natural amounts and then excessive and nuisance levels of filamentous green algae? One more slide about kind of field notes here. When we're in the streams and rivers, so one of the biggest ways that you can see from the riverbank if it's filamentous green algae is whether it has these long chains of cells, right? That's part of its name, filamentous green algae. It links together. Hopefully you can see this, and the podium isn't in the way, but this is actually water's net, which comes together and links together in like hexagons and octagons, which is pretty cool when you separate it out. And then we have also some different types like Cladophora that also is called mermaid hair, probably, made up by a little 6-year-old girl putting it on her hair, having long Rapunzel locks or something like that. I personally cannot stand it when somebody calls it seaweed. We're nowhere near the sea, and I love the beach as much as everybody else, but filamentous green algae is not the same as seaweed. Another really great indicator is the color of this. Again, kind of in its name, filamentous green algae. There's not really any blue or purple undertones. It can be very bright, verdant green, or it can be kind of a duller, I want to say dirty green, but that's not a really good description. But, it always has those green undertones. So we really want to look for that when we're in the rivers and streams. It's easy to pull up off of the stream bed or the substrate. So there's no root system. It's just kind of attached to bedrock, to sand, to substrate, to other plants even, to whatever's growing. I've seen it on top of snail shells and clam shells, that sort of thing. So one of the things that I read online, not that to say that this is completely scientific, but it's a great sort of really easy stick test, is to grab a stick and attempt to lift the algae out of the water. So if this mat of green stuff lifts up and sticks to the stick itself, and you can see the threads and the filaments, then that's a really good indication that it's filamentous green algae. If it slides off, if it's gooey and icky and more gelatinous, then that's an indication that you're not dealing with the filamentous green algae. So that's just a quick kind of field test there. All right, so what's the nuisance algae issue? How did this come about, and why are we so concerned about it? Well, it all started over a decade ago in 2010, where DEQ and EPA worked with the Shenandoah Riverkeeper and the Potomac Riverkeeper Network to understand, these reports of excessive algae. There was excessive growth. There was a lot of green stuff in the river. So again, what can we do to better describe this extent of algae other than just saying, gosh, there's a lot in there? So based on complaints and reports that were received by citizens, 5 different segments of concern were identified, and you can see them on this map. So there's 2 on the North Fork Shenandoah for For you all in the back, the colors aren't really vibrant here, but this side of the map, the Shenandoah County and northern Rockingham, is all the North Fork Shenandoah. So there's two segments here. This is right around the town of Timberville, outside of Broadway. Old Hill Cider, if you're a local cider enthusiast, if that rings a bell to anybody. And then the other segment is up around Strasburg and just south of that at a place called Deer Rapids, which is a fantastic boat launch, and I'll show you some pictures and you'll put it on your list of places to visit this summer. So we had two segments on the North Fork, and then we had 3 on the South Fork here. So this is the kind of yellowish color that's draining the eastern part of Rockingham County, Page County, through Warren County. So right around here is the town of Elkton, and then these two segments are getting very close to Andy Guest State Park. So this is the Rileyville boat launch, is kind of right around there. And then Bentonville, if you've ever done a canoe livery service on the South Fork, chances are you've been to Bentonville. So these 5 segments, in summary, were really where we were focusing in on our efforts. How can we look at the algae? How can we develop a monitoring method? And really, kind of getting down into that question of how much algae is too much algae. So again, with these 5 segments, we committed to follow-up monitoring after we listed them in the 2014 Integrated Report on Water Quality, which is actually required by the Clean Water Act. This is the place where every state in the United States puts together a list of waters that are meeting meeting water quality standards and those that are not. In 2014, we identified these segments to EPA. We said, all right, we're getting our act in gear. We're going to really understand the nuisance condition. In 2016 and 2017 field seasons is where we honed in on our methods. We were lucky enough to be able to work with a lot of other states, particularly West Virginia, as well as Montana and Colorado, who have already developed nuisance algae monitoring protocols. And so we were able to kind of look to their examples, understand what worked in their states, talk to them a little bit about maybe what didn't work, and really get a better understanding of what was going on there. I'd also like to identify the growing season. When we're talking about growing season, we're really talking about the time when people are out on the river. And for probably about 95% of us, that's May through October. So that's really the times when we're out looking for algae. We all know people that are outside of those norms, but again, that's kind of like the generalized majority of people. All right, so getting into our field methods, after we tried a variety of different types and different protocols, we really focused in on a 3-stage process. So when we were coming up with these stages, we wanted to make sure that we were basing this on quantifiable, scientific, and defensible methods that could be reproduced easily. They could be easily trained, that were relatively quick and easy, i.e. not taking 8 hours in the field, but maybe only taking 1 or 2. We really wanted to connect what we see from the bank to what's growing in the river to then a lab-derived objective parameter. And again, we were lucky enough to be working off of some of our other states. So I'm not going to give away the parameter yet, but just keep that in mind. So first stage is to stand on the banks and look at the river. What is looking at a transect, which is a imaginary line connecting one bank to the other in a straight line, so kind of an imaginary width line. What's the percentage of filamentous green algae that's covering the substrate or the bottom of the river? So we our staff are trained to come up with that estimate. We're actually very well calibrated, and I have some data to show you that's going to amaze and wow your friends. And then we also, from there, the next point is to take a Serber sampler, which I'll give you a shot of here in a minute, and collect the algae as it's growing off of the substrate, and clean it, rinse it, and then weigh it to understand how much algae is growing as kind of a quick and simple snapshot of that transect. And then the last stage is to take that weighed sample and to send it to our lab to be analyzed for chlorophyll A, chlorophyll B, and ash-free dry mass, okay? So chlorophyll A, oh, whoops, that was the wrong one. There we go. Chlorophyll A was actually the lab-derived parameter that we ended up focusing in on as what would eventually go into our water quality criteria. And that's for a couple of reasons, which I'll show you in just a second. But, wrong one. There we go. So just as a quick we already talked about how to identify filamentous green algae, but here's what it looks like in comparison. When we're looking in, we use an aquascope here, which is kind of like a tube with two pieces of plexiglass that enables you to see what's growing on the bottom without getting your face wet. I wish I had that when my kids were toddlers and in bath time and stuff, looking for all the bath toys underneath. That would have saved me a lot of time. But anyway, it's super helpful for looking at algae and what else is growing on the bottom of the stream, because we can really, when we're up close, looking at the frame of the Cerber sampler, what's growing in there, filamentous green algae, oh gosh darn it, there we go. Filamentous green algae looks a lot different than the blue-green algae, right? Remember I said the gelatinous kind of blue-green undertones? That's what that looks like. And then that looks different than our submerged aquatic vegetation or plants that have a vascular system, they have a root system, they're plants that really like to grow in the water. So they all look very different, and we're capturing what's growing in our as we place the Cerber sampler across that transect. We're really capturing all of this data, but what we're collecting is, as a sample to send to the lab, is just the filamentous green algae and the blue-green algae. All right. Now I want to go to the next slide. So here's a great picture of our Cerber sampler. It's basically a right angle frame. So there's one frame up here and one frame down at the bottom. You can see that our biologist Bill Van Wart has the frame on the bottom of the stream. This shroud is what they call it. I didn't come up with that name. But this is what actually we harvest the algae and put it into. So all that algae that's growing in the Cerber frame, Billy is scraping off of the bottom and putting into the shroud, and we composite it together because, let's show the next slide. So again, here's, as you can see, this is a sample where there's a lot of algae growing. This algae, we're just harvesting what's growing on the bottom, not necessarily what's floating up at the top, but what's growing from the bottom. So he's putting all of that in the shroud and putting it into the bucket. The fun fact, I'm going to tell you lots of different fun facts to amaze and wow your friends at parties. Transect is one, Cerber sampler is another. Another fun fact is that the area of this frame times 11 almost exactly equals a meter squared. And a meter squared is important because that's what all of the other states were using for their chlorophyll A values. So, the magic of math means that we could collect 11 samples of this, of the algae in this frame across the transect and come up with a very easily and quantifiable amount of algae that was translatable into these other criteria. So there's the sampler. So there's where we bring the frame up and we, all of the algae that's collected in the shroud is put into this composite bucket so we can mix it all together, we can rinse it, we can take out all of the snail shells and the clams and the dragonfly larvae and the crawdads and everything else. We can take that out and then come up with our algae. And here's what's left on the bottom. After we harvest out the algae from the frame, the bottom is just basically substrate. And again, we're doing that 11 times across the transect to get a meter squared total area. All right, so where have we collected data? We've collected data, like I said, we started with those 5 segments of concern that EPA, DEQ, and the Riverkeeper kind of agreed upon. We have since expanded in 2022. We expanded beyond, in 2021, we expanded beyond just those 5 segments of concern, and we chose sites based on complaints and reports that we had heard. We determined that we really needed to understand on a broader context where algae was growing in the basin. So we added 2 new sites. Hopefully you can see this from where you're at. So kind of the purpley segments are our segments of concern, the original 5, and then we've added a number of stations. We looked up here in Front Royal, just outside Front Royal, at a place called Riverton, if you're familiar with that. Seven Bend State Park was another location, the Lupton Road entrance. We also added Elkton's Town Park, that's right there on the South Fork. That was a little bit south of our original segment that was identified, but that's given us some great data. And then here's those 2 middle segments on the South Fork for Rileyville and Bentonville. So these are all the places where we have established monitoring stations. Again, we were visiting on a monthly basis, May through October, since 2017 is when we first started. All right, our second priority has always been to respond to complaints. When we get complaints sent in to my email address or coming in through the main line, we respond based on whether there's public access to that stretch of stream, whether there's recreational potential there. We focus in on the main stems of the North Fork, South Fork, but we also will go out into some of the major tributaries to collect as much samples and to get as much data as we can, right? We want to understand where it's growing all over. But of course, you know, resources and staff time is definitely one of our biggest limiting factors, which is where those college students, you know, will come in handy. All right, so we can't go out. Unfortunately, it's a little chilly today. I don't know how many of you would like to get wet and collect algae samples on a day like today, but maybe we'll just do a case study or 2. So I thought this would be a good way for folks to kind of see and understand how all the monitoring methods and the data that we collect come together. So imagine it's hot and humid. It's August 5th. Of last year, of 2021, 2 years ago. We're on the North Fork Shenandoah. We're at the entrance to Seven Bend State Park. It's a beautiful day, but gosh, look at that algae out there. There's some algae on the North Fork, and we need to understand how much there is. So our first step is to stand on the bank, view just what you're seeing up here, and try to get a sense of what the percentage of the substrate that's covered by algae is. If you were, again, to draw that imaginary transect line across the river here, our staff's visual observation was about 30%. At this time, we're lucky enough to have such trained and experienced staff that they can differentiate between the submerged aquatic vegetation or some of the blue-green that we see. So 30% filamentous green algae. We went ahead and we got our Cerber together. We threw it 11 times across our transect. So, you know, let's say that this is, oh gosh, just a randomly chosen number of 22 meters across. If we were collecting 11 samples, that would mean that we would be collecting every 2 meters. And so, you know, we collect that algae sample all together, and at the same time, we're looking at within the Cerber frame how much filamentous green algae is covering the Cerber, how much blue-green, et cetera, et cetera. And so for this particular example, when we threw it 11 times, the average estimate of filamentous green coverage was 34%. As you can see, that's pretty darn close to 30%. Got some Team Awesome going there. I'm pretty proud of that. When we collected that algae sample, we rinsed it, we took out all the bugs, we weighed it, and it weighed a total of 172 grams. That's like a stick and a half of butter, if you can imagine that. So that's a significant amount, maybe not a ton, but that's quite a bit. And then after we sent it to the lab, the chlorophyll A value there was 111 micrograms of chlorophyll A per meter squared. So that's a really good translatable number to work with our other partners in West Virginia, Montana, and Colorado as well. All right, so here's another case study. One more. Oh, whoops, I have some more pictures. I forgot. So here's another example of what it looked like. This one is a really good photo, this last one, because you can kind of see how there's some not filamentous green algae growing here. So this site actually had a significant amount of submerged aquatic vegetation growing as well, which kind of hosted the filamentous green algae. You know, it was attached and everything. So our overall submerged aquatic vegetation estimate for this site was over 25%, which is quite a bit, actually. And if you're thinking of the entirety of the stretch of the stream, you know, for about 35% to be covered by filamentous algae, and then another 25 to 30% to be covered by submerged aquatic vegetation is, that's, you know, halfway across the stream already. So, yeah, that's getting up into the significant portion. Do I have another photo? I do not. Okay. Next case study is at that place that I mentioned earlier. We're again on the banks of the North Fork. It's again August, so it's nice and warm. And this is at the Deer Rapids boat launch. If you've never been to Deer Rapids, it has a fantastic swinging bridge, which makes it really easy to get nice aerial photographs of the substrate and algae coverage of the stream. So anyway, this was again one of our monitoring stations. We went out, and as we were here, we don't even have to stand on the banks. We can go up in the swinging bridge and take an estimate of how much algae is covering the substrate of the stream. Our staff estimate was about 80%. When we threw the Cerber 11 times across the width of the stream, our estimate within the frame of the Cerber was about 78%. Again, we're getting pretty good at this after doing it for 7 years now. When we collected the overall sample of filamentous green algae, it weighed 690 grams, which I think is the second or the third heaviest biomass sample that we've ever gotten. So, you know, those of you who are familiar with translating metric into normal American, that's about a pound, over a pound, right, of algae. If you think of it, that's just algae. That's not the snail shells. That's not the dragonfly larvae. That's just the algae. That is significant. And then when we sent that away to the lab for our chlorophyll A value, that was 614 micrograms per meter squared. So again, that's a very high number and indicative that there's definitely a system kind of out of balance here. So I give you these case studies and kind of review a couple of these data points, but honestly, we could geek out on data all night long, and I would love to do that. But I know that we have other speakers tonight as well. So I wanted to say that if you are interested in getting into the meat and bones of the nuisance algae data, the 2024 Integrated Report on Water Quality, which is coming out next year, will include all of the nuisance algae data collected since 2017. And this is going to be assessed to new water quality criteria, which is in the final phases of being approved by EPA. So this is really the water quality standards, if you will, which will set in stone how we assess algae when we understand what is the nuisance level. So that's right now in EPA's court. Again, this criteria is going to be very specific to the Shenandoah. It will only apply to the main stem North Fork, South Fork, and main stem Shenandoah rivers. It will only apply to the wadable portions because, as you can see from some of these photos, we really need to be able to wade. Our best height we estimated is about 0.7 of a meter, but we have done it in up to 1.5 meters of depth. You just have to have somebody who can hold their breath for a long period of time to get down under there and collect the data or collect the algae and put it in the shroud. So all of this assessment, all of this data and the final assessment decisions for this criteria will actually, the criteria itself will be summarized later this month for the assessment guidance. That's going to be released to the public, like I said, later this month. Feel free to peruse it, and then the data itself and the final assessment decisions will be available next year. And I'm hoping that we can maybe do something like this again, or have a webinar or something to really review the algae data, kind of go over the issue itself. Oh, I had more pictures. and be able to get in depth with those folks who are really interested in that. So here's a couple more photos of what Deer Rapids look like. And so you can see how that algae really does cover the entirety of the substrate. It gets wrapped around your ankles, wrapped around your fishing lures, all that kind of thing. So our next steps for 2023, we're expanding our geographic range of monitoring yet again. We'll be adding a completely new site based on complaints that we've received. So, I still can't get this. So that new site is up here at Luray Landing, just outside of Front Royal. That's on the South Fork Shenandoah. And then we'll continue to go to Elkton, Riverside Park, as well as the Lupton Road Bridge that goes into Seven Bends. Those two sites we've monitored for 2 years. We feel like we really need to keep them on the list, keep going back, collect more data. And then our kind of long-term sites will be Timberville, and then the Deer Rapids boat launch with the cool swinging bridge that you all got to visit, and then Strasburg Town Park. So those 6 sites will be monitored, like I said, monthly starting in May. All right, so with that, do we want to do questions at the end? I want to make sure that we have lots of time to talk about Dr. Steffen too. So, at the end? Perfect. All right. Thanks, everybody. I appreciate your time.
Great. Is my mic on now? Can everybody hear me? Great. Okay. Thank you for the invitation to come and join Tara to talk about the different types of algae that we see in our watershed. I'm especially excited to be out this late. I have a 1-year-old and a 3-year-old, so I don't get out this late very often, and I think they're probably giving my parents a very hard time right now. So I'm happy to not be at my house. So I'm going to talk about a different group of algae that we sometimes see in different areas of the Shenandoah River and lakes and other rivers around our watershed and around the world, actually. And those are cyanobacteria, which are also known as blue-green algae, which can sometimes be harmful because of the different toxins or other compounds that they can produce. So I am trained as a microbiologist, and usually when I talk to different groups and I say that I'm a microbiologist, or I ask what people think of, whether they're a 5-year-old or an 85-year-old, when I say microbe, they almost always say the organisms that make you sick: bacteria, germs, gross, sneeze, whatever. So I'm actually a microbial ecologist, and that means that I study the way that microbes, whether that's bacteria or algae or viruses, interact with each other and their environment. And one of the things that I always say is that the vast majority of all of the different microbes that we can't see on the planet don't infect humans. They don't care about humans, but they do actually run the world because they're responsible for a lot of the processes that keep our planet going. So even though I'm going to be talking about algae that are problematic and cause blooms like the one that you see in this image here, please remember that not all of them are bad. So what we're looking at here is a bloom of toxic cyanobacteria in a small reservoir. This one is in Auburn, Alabama, at a site where I have a collaborator who runs experiments with me. And you can see that the sludge on the surface is thick and green, and I can tell you from experience that it smells awful. And blooms like the one that you see in that image happen all over the world. So this is a map of the entire planet, and it's not important that you can see what the individual countries are. Instead, what I want you to notice is that almost every single country on this map is either blue or green. And that means that in all of those countries, they have reported blooms of cyanobacteria, whether they're toxic or not. And what I want to note also is that in the few countries where these reportings have never occurred, it's probably not because they don't happen. It's probably just because they don't necessarily have the infrastructure or the resources to do so. And even in places like Antarctica, where blooms have never been observed or occurred as far as we know, we find genetic signatures of the organisms that can form them and also of the genes that they have that allow them to produce these different toxins that I'm going to talk about. So I have a bunch of images here to give you an idea of what cyanobacteria can look like in different places where I've collected samples before. So this first one is a video of a bloom in Lake Erie that I was sampling from in 2017 or 2018, I think. And one of the best descriptions I've ever heard of a cyanobacterial bloom like this is that it kind of looks like egg drop soup if it was really gross and green. So this one is in Lake Erie, and Lake Erie has a really long-standing reputation of being a lake that's plagued by pollution issues. In the 60s, one of its tributaries actually caught fire, and so that's why we have a lot of nice art renderings of the problems in Lake Erie itself. But this is a problem all over the United States. This is Lake Anna, actually this last summer, and you can see that the water looks very different. But we have been able to detect the genes necessary for toxin production, and there's monitoring of toxins that goes on in Lake Anna every year. This is a bloom in a Chinese lake that I study, and again you can see the green swirls there of the organisms that are forming the bloom. And here is the Potomac River and then the entire Chesapeake Bay watershed. So the Potomac River actually had blooms of the organism that I study the most in the 80s, but large-scale blooms like this one haven't occurred for a long time. So this image is one that a couple of students took in August at a site in the Shenandoah. I think that's that same bridge, right, that you were talking about? Yeah, yeah. Okay. So here are my students going to collect the samples that I sent them to look for. And you can see, we can see images just like the ones that Tara showed, but there were some other areas where we actually found the not so bright green and actually the organisms that might be responsible for producing toxins, the cyanobacteria or the blue-green algae instead. So my students collected samples for me and we brought them back to the lab. And before I tell you what we actually identified, I want to talk a little bit more about why algae are potentially dangerous. So these cyanobacteria can produce toxins and a wide range of other chemicals that can result in things like rashes and itching, which we call contact dermatitis. They can cause breathing issues and asthma attacks depending on your situation. And some of those toxins can actually cause liver damage or failure and have been implicated in liver cancer for people with chronic exposure. And some cyanobacteria can actually produce neurotoxins as well. So these can be quite dangerous. One of the most widely studied toxins is called microcystin. When it was first identified, scientists actually named it fast death factor because of how quickly it killed the mice that they fed it to in their research labs, to give you an idea of how potent this is. So this is a hepatotoxin. That means that it affects the livers of animals like humans, but those that are affected are actually usually not humans. But every year, there are reports of animal deaths in the United States and globally. So a couple of years ago, there was a large elephant die-off. Sorry, this is kind of hard to see. What that headline says is that toxic algae caused mysterious widespread deaths of elephants in Botswana. 300 elephants died because they drank contaminated water that had this toxin in it in Botswana. And then if you just Google the name of this toxin and dogs, every year there are pretty widely sensationalized stories of dogs that die from playing in or swimming in or drinking in water that's contaminated with these organisms. So you can see this is dangerous to large animals. If it can kill an elephant, it can definitely kill a dog or a cow or even a human if you get too much exposure. Now, luckily, there haven't been a lot of reported human deaths due to exposure to these kinds of things. You have to drink quite a bit or be exposed for a long time. But there has been some more notoriety. There was even an article in BuzzFeed, so millennials and Gen Z actually maybe have seen this, last year about how to protect yourself and your animals from toxic blue-green algae. So these are some large images of some filamentous samples that we've collected. These first couple here, you can see me swirling it around a little bit. Sorry about my messy lab in the background. This is what those mats, those cyanobacterial mats or filaments that I collected from the Shenandoah River look like now. We've actually been keeping them in culture, so getting them to grow in synthetic freshwater, basically, that we can make over time. And you can see that they have this dark bluish green color instead of that bright green color that Tara noted. And it's more, the best way to describe it is gooey instead of forming those nice, beautiful, long green filaments. Instead, it kind of forms this dark green, sometimes almost black looking sludge on the bottom of our flasks. This is an example of another filamentous blue-green alga that's actually a sample that we collected from Lake Erie last summer called Microcyra. So this one looks more like kind of a matted, almost like a wig or hair, but it does have that darker bluish color instead of being that bright green. So that's a really great way to identify the difference. And here is an example of actually some Microcystis, which is not a filament, but it's a different type of blue-green algae that I collected from a pond right outside my office on the JMU campus because we have blooms there every single year now. I think it was brought in by the ducks and the geese that fly in all the time. And I have actually identified the genes necessary for toxin production in that reservoir, and then from Black's Run, the little creek that runs out from that little retention pond from there. So we took the samples from the Shenandoah last year, and this semester, actually in one of my classes, we've been running some experiments to try and identify what they might potentially be. Because I am not as familiar with the filamentous blue-green algae, I usually work on those other ones, we wanted to do some molecular analyses to try and understand what those organisms were, because one of the first steps to trying to combat these organisms and their presence in a place like the Shenandoah River is to figure out who they are. And so we did a simple molecular test, and we identified them. We're hypothesizing that it actually belongs to this group called Limnothrix. So there's a lot of limited research done on this organism. Most of the work is done on the more conspicuous and charismatic organisms that cause large-scale blooms in important systems, like Lake Erie or in Lake Okeechobee in Florida, for example. This one, because it's filamentous and benthic, meaning it's on the bottom, it's harder to see, it's not as charismatic, and people don't pay as much attention to it, but it's probably just as important, particularly because it's in our own watershed. So it's only recently been discovered that this organism has the capability to produce toxins, and it might potentially be a novel toxin that we have not identified before. What we know, based on a series of different studies from groups from Australia and Brazil working in the Amazon and some other large-scale rivers, is that these organisms have the capacity to produce something that damages and can kill mice, including damage to their liver, to their lungs, and to their gastrointestinal tract, so their intestines, their stomach, their esophagus. And in addition, this toxin or a second one has some suspected neurotoxicity, which would affect the brain and other parts of the nervous system. And so I have some images here to give you an idea of what this organism looks like. You can see that it is this long filament here, and when it's grown in culture, it forms these kind of blue-green looking mats, just like the one that we see in our own cultures here. So my lab is actually currently working on some more detailed microscopy. My collaborator, who's also my husband, who is actually next week when he gets back from his course trip to the Bahamas, is going to be producing some really fine detailed images on an electron microscope of these filaments to better capture what their morphology looks like on a really fine scale. And we've done some more detailed molecular experiments that we're planning to get the results back from in a few weeks, actually. So I would be happy to share those further, and I can talk to Karen about how to do that potentially once all of those come back, to try and even better understand what these organisms are doing and what they're capable of. Now, I mentioned that I'm a microbiologist, so it's not just the cyanobacteria that I'm interested in. And in fact, one of the things that my colleagues and I have really been looking for lately is thinking about blooms of these organisms that form, not just thinking of the algae itself, but thinking about all of the other organisms that live with it. When you think about those mats of the green algae, or you think about these kind of gross, sludgy clumps of the cyanobacteria that are growing in the river, it's not just the algae themselves. There are also other organisms that find those algae pretty delicious, actually. And that gooey sludge that they're making, or those long, beautiful filaments, make a really nice home for other bacteria that could potentially live with them and interact with them. And so we know now that cyanobacteria and other algae actually have a microbiome, so organisms that actually live with them and support them. And they kind of interact to form a happy symbiosis with each other. That can be potentially problematic accidentally for humans, because some of the organisms that actually live with algae are human pathogens. So we've identified some gastrointestinal pathogens as members of the algal microbiome, organisms like E. coli, which can be potentially pathogenic, and organisms like Klebsiella, which also can cause issues, sometimes like pneumonia even in humans, so they can be potentially dangerous. So this is also something to keep in mind if you were to see a sign or a notice about avoiding an area because of a potentially toxic algal bloom. In addition to those algae, those blue-green algae being there causing the toxin production, there could be other organisms associated with them that could be potentially dangerous. And you can see, just based on some simple things that we've done, looking at these organisms under the microscope and then even plating samples. So we took the water from Lake Anna from last summer and we put it on a petri dish, and you can see nothing that grew on that plate is green. That's all bacteria and fungi that live with it. And you can see if you look under the microscope here, the vast majority of these samples have bacteria living with our large algal cells here associated with them. And in this image here, it's really striking because those few red balls that you see are cyanobacteria, and all of the green stuff is the bacteria that live with them. So it's just something to keep in mind that the algae don't live alone. And this is an example of the organism that I studied, Microcystis. This is what it looks like in that electron microscope. So we're hopefully going to have some images of the Shenandoah River samples in this fine of detail actually next Tuesday. I know. I tried to get him to do it before he left, and he wouldn't. So in my lab, I wanted to give you an example of what it looks like when we go out and collect samples, and it really depends on the size of the system in which we're sampling. So sometimes we go out on a huge lake like Lake Erie, and we travel on a Canadian Coast Guard ship or some other large research vessel, and we have to stay on that vessel for up to a week at a time to hit all of our different sites around the lake. Sometimes we can use an inflatable Zodiac to go in Lake Shenandoah or the Shenandoah River to quickly collect our samples, or I can just go on campus or send my students outside of my window, actually, to collect samples from the retention pond. And this is usually what our water samples look like when we collect them from an algal bloom, and obviously fresh water is not supposed to be that color. In my research lab, usually what we do is take those samples and digest them, doing some different types of molecular experiments to try and understand how the cells are interacting with each other and their environment, like I said. So a lot of our work is to understand how they use different nutrients that they get via runoff, or to understand how they're potentially interacting with bacteria that may be present with them to be successful. But overall, the primary thing that we're asking is, why do cyanobacteria bloom? What's happening to cause them to be successful in our environments? And why are they suddenly showing up in places where we've never seen them before? Is it just because we're paying attention, or is it because something has changed? And those are really the types of questions that we try and ask. And while most of my research has been in other locations previously, we're trying to do more at home in our own watershed. So I wanted to finish and move into questions just by giving you my own contact information and let you know that if you ever see anything that you want me to be aware of, and now that I've met Tara, she and I will be in contact also, I would love to know if you have a sighting that you think that I would be interested in looking at. Feel free to email me. I would be happy to see pictures or locations or anything like that. So thank you.
Thank you, Morgan, and thank you, Tara. We will now do some questions and answers, and I would encourage you to wait for the microphone because we do have an online audience as well. So both Wayne and I will be walking around with microphones. I'll turn that one on for you and take this.
Okay.
I understand that you're characterizing the areas where the blue-green algae is found. Are you collecting any data like the nutrients in the water, or is your lab looking at what causes these blooms, or are you just focusing mostly on understanding what the algae are?
Is that for me?
I guess, yeah.
Yeah, okay, yes.
I mean, I would probably ask the same question of both of you, but
Yeah.
I don't want to take up all your time.
So yes, in my lab, we always try and collect the sort of environmental data is what we call it. But things like nutrient concentration, we usually focus on different forms of nitrogen and phosphorus specifically, because we know from several decades of research now that those two are some of the most important for cyanobacteria. Also things like water temperature, conductivity, turbidity, so how well the clarity of the water. We're trying to start to look at toxins also, but yes, we do collect that information.
And when we collect our nuisance algae data, we're collecting field parameters, which is pH, temperature, dissolved solids, dissolved oxygen. So we're getting a very quick snapshot. Most of our nutrient data is collected at our long-term trend sites. And so those are stationed throughout the Shenandoah Basin, but not tied directly to the point at which we're collecting our nuisance sample. But we do have long-term data sets for nutrients as well.
Do either of you have done research on the effects of climate change as far as algae blooms, or know of studies that have done that, and what results might be coming?
I'll go first, and then I'll let Morgan answer. For us, just in the past, 6 years that we've been looking at nuisance, we can see definitely how it relates to weather, and we all know weather isn't climate. But for instance, many of you remember 2018 as the year when it did not stop raining. We had hurricanes, we had pass-throughs, we had late nor'easters, and so we really did not see the algae growth that we had seen in previous years or since. The river was just so high, precipitation was so high, flow rates were up, the hydrologic stage was incredibly high, so we didn't see algae grow that year. But in other years, in very short time spans, when it's very dry, very hot, when those rivers retreat to base level or below even, is when we really can see algae take off, especially the blue-greens. So I don't know that we're enough along to really talk about climate change, but we can see how it's fluctuating in a short period of time, and how conditions, depending on the effects of climate change in the Shenandoah Valley, could be pretty drastic.
Blue-green algae in particular are better able to outcompete other types of algae the warmer it gets. I have a colleague actually who has made his whole career on the phrase blooms like it hot, because he was the first one to sort of publicize and do a lot of the work for us to understand that cyanobacteria generally do better when the temperature increases. The other thing is there are some filamentous cyanobacteria that can produce some pretty scary neurotoxins that have been equatorial, so usually they've only been in tropical environments, that are now not only just pervasive in the south of the United States, like Florida, but we also now have found them as far north as the Great Lakes. And so these organisms are expanding to areas where they haven't been able to be previously, because it is getting warmer, or the warmer temperatures have allowed for them to be able to better survive and even thrive in temperate northern systems where we would never have found them previously.
Question over here. Hey, Tara. Mark Frondorf, Shenandoah Riverkeeper. Thank you for this presentation. Really enjoy it. I'm happy to see that you guys are expanding the number of sites that you're going to be collecting samples from with respect to chlorophyll A. But, 7 sites, that's how many total I think it's going to be, you said?
6.
6, okay. 6 sites. So that comes out to be, 100 miles on the North Fork, 100 on the South Fork, roughly 50 on the main stem, maybe like one on average, one every 40 miles or so. What are you guys doing to check on the algal blooms that are occurring that aren't close to these 6 sites? Because, we patrol the entirety of the river, and oftentimes we see algal blooms that may be in the middle of nowhere, but they're still in between point A and B, where people are recreating, either swimming, fishing, tubing, or whatever on there. And then the second question that I have, is there's the $2.5 million 3-year harmful algal bloom study, and I was just wondering if you could comment a little bit more about that.
Absolutely, yeah. So, Mark, you make a great point. Did I mention I'm hiring this summer for that extra help? But no, you are exactly right. We wish that we were able to add monitoring stations, but for right now, 6 is kind of what we can handle with our current staff level, knowing that we have so many other priorities, and we want to make sure that we're able to respond to complaints and reports when we receive them. So yeah, complaint response, even though, you know, in my presentation it was the number 2 priority, that's still an important part of our work. And as I hope demonstrated, you know, it informs our site locations for the future. So we've been able to use those complaints and reports to change where we're monitoring and where we're collecting data. But yeah, it's a tricky situation, and we'd love to add some more there. In terms of your second question, for those who were not aware, in the 2022 legislative session, the General Assembly prioritized $2.5 million to study harmful algal blooms on both Lake Anna, which was in Morgan's presentation, as well as the Shenandoah. We've been able to kind of separate the 2 projects because they're such different systems, really different issues completely. So the Lake Anna study will feature a lot of data collection from both DEQ scientists as well as USGS scientists. In the Shenandoah, it will be a broader network of partners. This is still under construction at the moment. When it is made public, we'll be able to post it on our website for everybody to see and read through. But I can tell you it will combine partners from USGS again, as well as the Interstate Commission for the Potomac River Basin. If you're not familiar with that quasi-governmental group, they have a team of fantastic algae scientists who've been working in West Virginia, Virginia, and even Maryland and Pennsylvania to collect data. And our attempt is to really tie real-time monitoring and create a surveillance network and collect some of the data that Morgan highlighted, taxonomic and genomic data for what algae is growing in the system, as well as the environmental data of water quality. What's the total phosphorus at this point? What's the nitrogen concentration at this point? So tie all of that together with algal blooms and create what we haven't had before, which is really a surveillance network, not just incident response of getting a call from someone saying they're seeing something green on the river, but actually being out there on a regular and routine basis with a diverse team of scientists to collect as much data as we can. So great question. Thank you.
Thank you. I'd like to thank the speakers again for their presentation. We really enjoyed it, but I'd like to, my name's Hal Ostlist. I'm with the Center for Cold Waters Restoration. I'd like to ask you to look at the other side of the equation, or maybe to complete the equation. As a card-carrying member of the Friends of Photosynthesis, a group I just made up, you mentioned earlier the importance of oxygenation that chlorophyll brings about. It also, remember, and for everyone in the group, I want them to remember, it fixes carbon. It reduces carbon dioxide to carbohydrate. So algae may have some problems with us in terms of toxic issues here and there, maybe unsightly if we're on a picnic with our best friend or what have you, but worldwide, algal production of oxygen and reduction of carbon dioxide is vital to our lives on Earth. Thank you.
I'm glad you said that. 1 of every 2 breaths you take is actually, the oxygen is produced by cyanobacteria in the oceans.
So you've I guess, a nuisance filamentous algae is, we know it when we see it. It's sort of like the definition of pornography. But so what is a healthy, what is the chlorophyll A for a healthy system?
That is a great question. And we're hopeful that as we go through this data, now that we have, that EPA is reviewing for us, and hopefully we'll have in our hot little hands sometime soon the level at which it's excessive. You know, anything under that would be considered the normal range. And so, sure. Yeah, absolutely. Probably under 100 milligrams of chlorophyll A per meter squared. So under 100 would be a normal amount. In a very broad range, I think it really does depend on the system and the ecosystem. You know, that's kind of some of the ballpark figures that we're determining for the Shenandoah Valley. It's a little bit different. Those numbers are kind of toggled a little bit different in West Virginia and in Montana. But that's very generalized, and we'll have more detail when it's released later this month in the assessment guidance. And you can get into again, we can geek out all day long on this kind of stuff, but you can really get into like the nitty gritty of what it means and what too much chlorophyll A in these systems is, both like as a one-time thing in the season, as well as a chronic issue that we're seeing throughout the season. So.
Maybe time for one or two more questions. Anybody? I have a question for Morgan. I know you mentioned about the toxicity of these blue-green algae for elephants and dogs, and I'm just wondering about fish.
Well, I think it can be problematic I think it's mostly toxic to mammals, but I'm not 100% sure. Generally, the fish die-offs I don't think are due to microcystin. I think it's due to the consumption of oxygen that occurs when a bloom dies. So bacteria will degrade all of the algal biomass when the bloom dies, usually at the end of a season. And when they do that, their respiration ramps up and they use all the oxygen in the water and they create an oxygen dead zone. And then you often get fish kills due to that instead of the toxin.
There's a question in the back there, maybe Tommy, and then we'll wrap it up.
In the case of that die-off, would you see those masses floating to the surface in like stiller pools? I've noticed that on the Shenandoah River when I've been out with clients fly fishing. Really gelatinous, brownish, coming from the bottom. You can actually see them releasing from the bottom and floating to the top. And absolutely no fish in those areas.
Oh, really? I don't know for sure what that would look like, especially in a river. I'm more familiar with what it would look like in a lake. That's a really good question.
It's late season when I've seen that, usually late summer, early fall, and it's usually real slow sections of the river that, you know, but...
we could probably figure it out. If you see it, you can contact me.
Definitely.
Mm-hmm.
Right.
Yeah, I think I know what Tommy's talking about. I've seen that too, where there's almost like flakes that peel off the bottom, and because they haven't trained gases, they float to the surface. So we'll let her know next time we see something like that. So please join me in thanking our speakers once again. Also want to remind you that the Friends of the Middle River have a booth out in the lobby if you want to learn more about that organization. Wayne, I don't know if you want to add any comments there.
We're just going to make a pitch if you didn't, be sure to stop by our booth out there.
And then for those that are following the project with the Virginia Museum of Natural History putting a branch campus here in Waynesboro, I will be at the public library, Waynesboro Public Library, on Friday from 4 to 5 p.m. with open office hours where you can come and learn a little bit about the progress there and ask any questions that you might have. And so with that, thanks for coming tonight, and I hope to see you again in April when we have our final talk for the season.